Okay, hello everyone. Welcome to another ISAC webinar. I'm very excited with everyone here joining us today. So today's webinar is on Introduction to Adaptive Clinical Trial Design. I'm Nusaiba Abdul Rahim and I will be your moderator today. I'm a senior lecturer and currently head of Department of Clinical Pharmacy and Pharmacy Practice at Faculty of Pharmacy, University Malaya in Malaysia. So before we get into the webinar, I'd just like to introduce to everyone here on ISAC and ISAC Early Career Working Group. So International Society of Antimicrobial Chemotherapy, so ISAC, it's a major society and it aims to advance the education and the science of therapy of infection through the establishment of an international federation of affiliated national and regional societies with similar aims and objectives to its own. So ISAC has more than 90 member societies and more than 50,000 individual members. So if you have any questions about ISAC, if you want to get more information, just reach out to us. So with ISAC Early Career Working Group, so ISAC has more than 20 working groups and ISAC Early Career is just one of them. So it aims to serve as a platform to communicate with senior researchers and provide young researchers worldwide opportunities to build long-lasting networks for collaborations. So here is the leadership team. And again, if you guys are interested to join, you can scan the QR code. So we often have discussions with other early career researchers, always happy to, get new ideas, get new suggestions on activities, events, and programs for early career researchers. So this webinar is actually the second webinar for the series on when pharmacometrics and systems pharmacology meet infectious diseases. So with this webinar series, we aim to share the latest research, the applications of pharmacometrics and systems pharmacology in infectious diseases. So as mentioned, today we'll have our second one on Introduction to Adaptive Clinical Trial Design, and there'll be many more to come with really amazing speakers lined up. So before we get into the webinar and before I introduce our speaker, just letting everyone here know that there will be a Q&A session at the end. So after the talk, we will have a Q&A session. So feel free to type in all of your questions in the chat box, and then we'll get to it after the end. So with that, it gives me great pleasure to introduce our speaker today. So we have Dr. Xiao Hui Amanda Wong. She's the Director of Pharmacometrics at Satara. So Dr. Amanda Wong has been working and researching in statistics, psychometrics, and pharmacometrics for the past 20 years. Dr. Amanda Wong started working in population pharmacokinetics, pharmacodynamic modeling, and exposure response modeling since 2016. Before that, she worked at PRA Health Sciences as a biostatistician for 4 years, leading studies of all phases and providing consultant for the pharmaceutical companies. Prior to her time at PRA, Dr. Amanda Wong was an assistant professor of statistics department at University of Virginia, where she has been teaching, researching, and providing statistical consultant service for UVA Medical School. Dr. Amanda Wong is an expert on mixed effect models, Bayesian statistics, and resampling techniques. So with that, let's all welcome now Dr. Amanda Wong.
Yeah, thank you very much for the nice introduction. And it's my pleasure to give a talk here. Thank you for the committee and for everybody. So I'm going to start my talk. So it is so let's share my screen. So, okay, so I said this is an introduction to the adapt clinical trial design. It's really a big name, okay? So it's there are so many types, so many different designs. So this, I hope, is serve the purpose to give you some idea why we need adapt design and how this is different from the traditional one. And I'll give a couple examples at the end. So the sorry, I got a cold, so I have a horsey voice. Please bear with me. So the traditional design, we know you have the plan. So then you go to you design your trial, and then go to the conduction. Then the database logs. Then you do the analysis. So this is a traditional. The adaptive design is during the conduction period. You're going to review the data based on the current data collected. Based on the current collected data, you're going to do some adjustment to your design. No, not to a design. Do some adjustment. Then you go to the conduction. This circle could happen many times. Okay, so usually the discrete cases we call it is through the interim analysis. Then now here's some continuous method. So it could be go as the conduction is going. So this is the major difference from the traditional design. So the goal is clear. You want more like a flexibility for the investigators. But this is it said, you review the data, then you adapt. Doesn't mean this adapt could be done in any way you want. Actually, all the adaptive changes is planned at the very beginning of the design. So here the condition, the first one is very important, is pre-planned. Okay, so it's pre-planned, and because you look at the data during the conduction. So this is here you cannot undermine the validity and the integrity of the study. Validity means you have assured the original research goal is not changed, and like you have to provide the correct estimation method to produce the meaning, correct, confidence interval, estimate of the treatment effect, and provide the proper p-values. So the integrity is like you cannot compromise the data and the trial, because you're going to look at the data during the conduction. The one thing is important is you cannot leak any informations. So this is the conditions. And another important is when you do the adaptive when you have any adaptive changes in your conduction, this is based on the data you collect in the study. You cannot based on any external data information. So this is how this is the conditions of the adaptive designs. And the change could be majorly is to the trial procedure change, like the study dose changes, you add a new arm, or like the study population change. And another is a statistical procedure change, like how you randomize, your your arms and like sample size, so on. So this adaptive design was for me, I probably focus more on statistical procedure change there. And another so those like adaptive design in statistics was like was first seen like 40 years ago if you look at the literature, but in the recent like 10 years it becomes more popular, because like 2004 FDA released this the critical released this critical path initiative, and then later I think 2006 FDA released this critical path opportunities. So basically encourage investigators to submit the designs of the adaptive design. The reason is during that period, before that, FDA found there are few submissions of NDA. So because the develop the drug, it's very expensive. how are you going to classify? There are lots of ways, but I just put the simple ones. The first is based on purpose. Okay, so they have adaptive randomization design. Basically, you change your randomization rules. Could be depends on, here I give example, response adaptive or covariate adaptive, depends on the covariate. Then it's like a group sequential design. So the traditional 3++ design, actually it is a group sequential design. And the sample size re-estimation, then it's like the drop the looser design, or drop one of the looser arms. So, and there's some adaptive dose finding design. So this one is the previous ones are all like this one is actually a continuous adaptive. So you change, almost it's not predetermined. It depends on each patient's assessment, and then you re-estimate their arms and dosings. So this is pretty new. Not pretty new, but this is like the methodologies in the early probably 1991, 1992. But we saw this more and more in the oncology design, the phase 1 design. There are biomarker adapt design, adaptive treatment switching design, hypothesis adapt designs. So then this adaptive seamless phase 2 to phase 3 design. The last one basically is any combination of the above. So the red highlighted ones, the ones I have seen, and it's like more common. The others I never, I didn't see that many designs, but I'm not aware any like new drug design through those ones. But I think group sequential and the first this 4 highlighted, they are actually the first 4 highlighted I think quite often, and the seamless one is also very often. If you based on the rules, you can classify the adaptive design into like 4 different type. So first, the allocation rules, how the subjects will be allocated to the arms. And the sampling rules, like you're going to re-estimate the sample size and include the different number of the subjects into your study at the later stage. Stopping rule is the one I have seen quite a few. Is how you're going to stop the trial early for different reasons, like could be for efficacy and for futility, for toxicity. So this is the third one. The last one, basically, like the adaptive dosing finding design. It does not fall into any of the first 3 categories. So it's basically, that's the decision rule, is the last one. Okay, so before I go there, before I give any detail of the design, I'm going to, So I'm going to talk a little bit about the statistical concept of the hypothesis test. This one is the fundamental one for most the phase 3, probably most of the phase 3 design, because at there you want to confirm whether there is a treatment effect. So the treatment effect is better than the placebo or standard care. So this is the whole thing is carried through this statistical method called hypothesis test. I'm not sure how many of you familiar, but I will go through some basic ideas here. I hope, because this I will talk talk later the examples based on that. So so the idea is the hypothesis test setting is always like that. So it's like the null hypothesis is they're same, and the treatment effect and the placebo are the same, and the alternative is the could be treatment is better, or treatment effect is different from placebo. This is what we call it one side or two side, depends. So this is the so here, when we try to make the decision, right? So basically we want to, based on our clinical trial, to make the conclusion the treatment is better than the placebo or not. But the truth, we never know, right? The truth could be they're same, or could be they're better. That could be the treatment is better. So there's like a basic four scenarios. Either we make the right decisions under the different choose, and who make the wrong decisions. Okay, so this exactly translated to the hypothesis test. So here, we same thing, like if we have a mirror to mirror transformation. Sorry. So here, the first one, we couldn't make the decisions to fail to reject H0, or that's the decision you're going to make, or the reject H0. So it depends on what the choice is. Unfortunately, we never know. So you could make the right decision, you could make the wrong decision. So the whole goal here for the clinical trial is try to control the both type 1 and the type 2 error. Okay, so this is So this is how this hypothesis test is about. Now I want to make a couple things here. Is like so first, I review lots of statistical trial protocols or analysis plans. So you will see the null hypothesis is always include the equal sign. That's how the mathematically, statistical theory is behind it. I did see some protocol, they said H0, the null hypothesis is treatment bigger, strictly bigger than placebo. If you see that, that means, they didn't put it right. It cannot be done. So this is the fundamental. Everything in the hypothesis, later derive those formulas. It's based on the assumption they're equal. Okay, and another is here you fail to reject the null hypothesis. Doesn't mean the treatment is equal to the placebo. Actually, it's just we don't have enough confidence to say they are different. So this is a pretty important concept here. Okay, I'm going to go through a toy example and to explain, here the sample size and how to control the type 1 error, type 2 error, how they related to what we know, like alpha or power here. So assume we know the truth. There's the treatment and this placebo. Okay, so this is a very simple examples, but the whole idea could be carried to any any hypothesis test. So we know there is a difference, the truth. Now the traditional, let's start with traditional randomization controlled trial. So we have 2 arms. Now here we just, each arm we have 5 subjects randomly, take 5 subjects from each distribution. Now here we have observed the values, right? So here is the placebo, have 5, and treatment have 5. Now we have the sample mean based on those 5 subjects. We have the treatment, the sample mean of the treatment, and we have the sample mean of placebo. Now there is the difference. Okay, so you probably can tell, okay, we're going to base on the difference of this sample mean to make our decisions. So, in our trial, we only have one. We always, we only have one trial. So we either make a wrong decision or right decision. So So how are we going to know the truth? Well, because this is a toy example, I could do simulations. So I simulate 2,000 trials. Each trial, I have 2 arms. Each arm have n subjects. Okay, so we have 2,000 pairs of those sample means. This is the xt bar and the xp bar. What do they look like? That's how they look like. So the histogram, the red histogram actually is 2,000 sample means of those of those simulations of the placebo arm. Each arm have like 5 subjects. So this is the histogram, and the blue one for the treatment. Same idea. Now those solid lines actually is the theoretical distribution we're going to base on actually. So this is the theoretical distribution. So they're pretty good. They should be. And those vertical lines, that's the one trial we observed. Okay. Now here you look at this, is what I'm going to, how I'm going to test they're the same or not. We know they're from the different, but look at their, I don't know. If they're one is like this side and the other is this side, probably we have more confidence to say, oh, they're different. Now, how we're going to make decision based on that. So this is actually, we talked about it. It's based on difference of this sample means, yeah. And then now we have to standardize in some way to make sure every every test and everything every test is it's comparable, right? So this is a test statistic, we call it T, and in our case we have observed the test statistic based on that trial. So based on the trial I picked up, so this is the test statistics of 1.3. Now the previous case, you can see the difference is 0.05, but I'm going to standardize it. Now this S pool, this means like how you're going to estimate your standard deviation for your distribution, or the original distribution. So this is also an important one. You need to estimate the to get the test statistics. Okay. So the next one is under the assumption, as I said, under the assumption they're the same. There is a theoretical distribution for this test statistic T. It actually is a T distribution. This is 2 sample T test comes from. And for our case, I put this T distribution with a degree freedom 8. Now this is how we make the decisions. So the red vertical bar, we call it a critical value. What that means is at the right side of this area of this vertical red bar, the shaded red area actually is equal to the significant alpha. Okay, so this is usually it's the 0.05, whatever the your alpha is chosen for your trial. Okay, now this triangle, blue triangle, that is the observed test statistics. And this shaded blue area on the right side of that point actually is the that's the p-value. That's the p-value we calculated. So the decision is we're kind of compare the observed statistical value is at the right side of this vertical red line or not. Now, in our case here, we're going to reject the H0. Basically, we don't have enough confidence to say they're different. These two these two treatment, the treatment and the placebo are different. Another way is using p-value. So you can see the p-value actually give you more information. Why? Because the p-value equal to 0.04, then it will be just at the right side of a vertical line, compared to some p-values of 0.0001, which is here. So the smaller p-value actually shows much more overwhelming evidence. Okay, so this is the idea of doing the test. So now, unfortunately, we have, the result. If we based on this data, now based on the one trial, our claim is we don't have strong evidence to, to reject our null hypothesis. Means we don't have strong evidence that the treatment effect is better than the placebo. Okay, so this is actually, this is the case with each arm of 5 subjects. Now, assuming we have the same difference, the same sample mean as previous with the 5 subjects. The difference here is the same, but instead of each arm has 5 subjects, now we increase to 20 subjects. Now, this is a similar idea. This is the distribution of the sample means. Now, because we have much more sample there, so you can see we have a better estimate we have a better estimation for the sample mean. Why? Because the standard deviation is much smaller, right? This is a much narrow distribution than the previous one, with each arm have 5 subjects. Now you probably can tell with the same sample mean we observed for that trial from that one, actually we can tell there is a difference. So based on the hypothesis test, now this is the same idea. So this is a critical value. Now this is observed value. So the p-value here is really small. And now we're going to say, oh, there is, evidence. There is evidence to say the treatment effect actually is better than placebo. Okay, so here we make the right decision. The previous, we make the wrong decision. Now this is a toy example. So I cook this example this way, showing sample size matters. So probably this can give you some idea. Sometimes you go there, the trial, you make pre-assumption of this, standard deviation of your population. Then later, you're not sure. So even you look at the literature, everything, you're not sure. So you're going to re-estimate the sample size based on the current data. So that's the, you can tell how the, re-estimated sample size works. Okay, go back to this concept again. So how to compare the type 1 and how to control the type 1 error? Actually, it's relatively easy. It's set up the alpha, okay? How you are confident of it, how you want to set the alpha. So alpha in the early phase, like phase 2, probably we put 0.1, but 0.05 is quite common at the phase 3 if it's 2 side. One side you use 0.025. Usually that's the that's the alpha. So based on this setting, we can control type 1 error. Okay, it's kind of easy. We just set up our significant level of the alpha. So I want to say one thing is that, some people always asking, okay, I only have one trial. I did this, I observed the data, I did all this, follow this procedure, I make the conclusion. Either it's right or wrong. So what do you mean you have this control the probability of type 1 error at like a 0.05? Yes, you're right. You cannot, for one trial, that's it. Either you make right decision, you'll make a wrong decision. But you think about if you do this all the time. For example, from FDA's point of view, basically that means FDA approve so many drugs a year, let's say 100. They control this type 1 error means they allow 5% to be wrong, right? So the 95% of them are right. So for one trial, this thing is this whole theory probably, you know, you say, okay, what's the meaning of that? But if you do it again, then the guarantee, it's not guaranteed, but you have like 5% to make the wrong decision. Okay, so this is the type 1 error. So type 1 error, it's easy to see here. It's strict related, it's strict just the significant level. But the type 2 error is hard to control. So here's the type 2 error. Because it's under the assumption your treatment is different from the placebo. So the control type 2 error actually is related to the power. That's we're talking the power. Now to calculate the power, it's not that easy. You need to put the detailed difference between mu t and the between the sample mean of treatment and between the treatment effect and the placebo effect. And you need you need based on this hypothesis test the setting as well, and you also need some detailed information of the difference. So actually the power is equal to, so once you calculate the power, actually the power 1 minus the power actually is the probability of the type 2 error. So usually we want our powers bigger than 80%. That means we want to control our type 2 error under 20%. So this is the idea. Now I just show one of the power here. So based on the current example, so let's see, you know, this is our true treatment effect is 2 and the placebo is 0. Now then I calculate the power. Okay, so the power based on basically is based on this exactly difference, and the powers looks like that. So you can see when sample size equal to 5, the power is about 90. So when sample size equal to 20, the power is very high. So you can probably tell I pick up this I pick up this simulation and make sure this is it's giving me this results, right? So the power, you know, basically we made a wrong decision where we're really falling into that 10% there. Okay, so this is the idea of so this is the whole hypothesis testing, hypothesis test idea. So now I'm give you a couple examples. I have 3 examples, but I'm not sure I can go, you know, through all of them. I'll give the first 2. If, you know, if we don't have time, just skip the third one. So the first one is a group sequential design with the stopping rule for efficacy. This is the Merck's immunotherapy. It's the we call it pembro. So the pembrolizumab. So the pembro is this. So this is the kind. I just you know, copied this from their protocol. So this is the phase 3 study, and then they have 2 arms. And this is predetermined. So this is the 2-to-1 ratio. We same, you know, one with the immunotherapy combined with the chemo. The other is just chemo itself. So the primary endpoint. This is a typical primary endpoint for the for oncology. So there's a progression-free survival. Now they have 1 interim analysis. This is ADAPT design. They have the 1 interim analysis. So they hope the trial stops if this pembro combination with the chemo, you know, it's much better. It's better than just the chemotherapy. The one thing is that they know their drug very well. The phase 2 showed pretty much the information the drug works. They do this. What do they want to do this? Because they want to get the early, you know, get the early results and get the early approved, and they could put it on the market earlier. So so here I want to see one thing. Now we talk about a significant level or type 1 error, the alpha, right? Now this alpha is for 1 trial. Okay, it's not for 1 test, it's for whole trial. So here, the group sequential idea, the one thing you probably heard is I for spending. It's like, because for one trial, you have this alpha 0.05. Now, how are you going to spend them if you want to look at a couple of times? You cannot do this like the first time interim analysis. I look at this, and I want to use this 0.05, and later maybe you have second interim analysis. I want to look at the test and use the alpha 0.05, and at the end final. Now you cannot do that. If you do the 3 times, actually the whole type 1 error for your trial is bigger than 0.05. So you have to control this. So you have to control the whole trial with alpha equal to 0.05. Okay. Now what they did, they have the one-sided test, so they control this alpha is 0.025, 2.5%. Okay, then they have one interim analysis. Now, how are you going to split this? This is lots of different methods, also depend on how you think about your study, other informations. So, this is you need to, go to a statistician to talk to them and discuss, how you're going to split this alpha. So here, what they did is the interim analysis will be conducted after 50% of the, not 50% of the subjects enrolled, it's 50% of the subjects, they already have the PFS results, okay? So you can think about at 50% of the subjects who already have the results, actually you have much more subjects enrolled. So this is important. It doesn't mean you, usually you have more subjects enrolled. So here, when to look at the interim, when to use the interim analysis is tricky because you have to calculate, you have to do lots of simulations based on the different scenery, because you don't want to do too early. Too early, you will not, the sample size is not enough. You might not see the results. You don't want to do it too late, but because it's almost close to the end, then why don't you just go to the, the end of the study? So what they did is like a 50%, and they use this, use the Landmatz, Oberyn, Fleming, spending function. You calculate this efficacy boundary. So what they have to emphasize is in the interim analysis, the test for the significant value, the test for the significant level is 0.0015. Okay, so they know their drug is working, and they put this significant alpha level. It's really small for the interim analysis. And then if it's not hit, they keep going. At the end, you cannot use 0.025 anymore. So this is 0.0245. This is at the end, if you do the hypothesis test, that's your significant level. Now, somebody might notice you add these two together, it's not 0.025, right? It's 0.026. So why you can do that? Because here these two tests, actually you use the data, they're correlated to each other, right? The second data set, the final data set actually have half actually is you used it before. So because of this correlation, you can adjust this alpha, make sure by this kind of adjusting the method, you can make sure the whole probability of type 1 error actually is still 0.025. Okay, so this is how this Pemboy study did. So this here is clearly stopped for efficacy, and you split the alpha and you consider the correlation between these two sets of the data, in the interim and the final. Then you can use some statistical method to do that. Okay, so another example is more complicated, but this is a Cape Cod study. They want to, the original is not, and this is for the pneumonia, and however started at 2015. However, later the pandemic started, and they have to involve more. They have the COVID infected patient involved. and that there is this urgent to estimate, for the COVID patient. So they have to make some change. So the decision is you're going to embed a sub trial, the Cape COVID, in the current ongoing trial, Cape Cod. And there's lots of methodology adjustment. First, the primary outcome, changed. And at the first, the original trials at the day 28, now is at the day 21. Okay, so this is a major. This is one adjustment, and the Cape Cod is the Cape COVID is a trial is first is being blind, and later they hope they could convert it to open label if the drug works, if the drug works. So they try to choose this group sequential design instead of the subgroup analysis. The reason, the first is important is like the endpoints are different. And secondly, they don't, at that time, they don't understand this, the infection pathway very well. So, and the third is like, okay, your standard deviation for the population at the very beginning is, based on the pneumonia, right? Now here you could have people got a pneumonia, got COVID again. So the population distribution standard deviation definitely, will be changed. So that's another thing they they don't want the subgroup analysis. They want the different design, the group sequential. And if you use subgroup, you have to wait till the whole Cape Cod study stops. So that's another reason. So they choose the group sequential, and they also, what they did is they want to, and they have to re-estimate the sample size, because as I said, the sample size, the power depends on the difference of the two treatment, the details, right? Now here, the original, here they don't know in the control arm, they don't know even in the control arm the failure rate of the COVID patient. So they re-estimated the sample size. So there are lots of changes here. At the end, I want to go, it's like, this is a group sequential, and they also keep 5% is the type 1 error. They want to do like a 6 interim analysis, because they really wanted a quick decision. And they keep this 5% of the type 1 error. And they use the rule combine the efficacy and the futility, and they use a very conservative efficacy rule, but they use a very aggressive stopping boundary, the stopping, for the futility. So that's how. And another reason is to use this method is simple, because they need a quick simulation. And then the simple, because you could train your investigators very well quickly, and they have already have a available package to do that. So those are very important when you do the operations. Now I want to show the final. That's how they got there. So here is the you can think about this as a test statistic, and when you calculate there, when you look at the 50 subjects, that's the first interim, and you can see if you test the statistic above this area, above 3.66, that means your treatment is it's significantly better. Now, if you're below this minus 0.91, that means you're going to stop the trial, the futility stop rule here. If it's in the between, you keep going. So this basically tells you, you're going to do at a sample size 100 or 150 like that. So this is the efficacy This is the stopping rule for both efficacy and the futility using the group sequential design. Okay, so this is the next one. I'm not going to talk about it. This is the design. Actually, we at the start, we helped the client to do that. This is the CRM, the Bayesian adaptive design. I saw somebody was going to give the talk later. That's great. So I'm going to skip this whole example. So here's some summaries. There's a good part of the adaptive design. So this is a really benefit of that. But there's a trade-offs and issues. There's lots of challenges. Hope everybody fully understands that. And because it's this is because the adaptive design usually is complicated, and you have to do lots of simulation to make sure you cover the worst scenario and the different situation to understand it better. Because I have done this a couple of times. Every time if it is an adaptive design, it will be scrutinized by the FDA for sure, because they also want to make sure they understand the design. Usually this is done through lots of simulations. Okay, so I list a couple of references there. I think I'll just stop here. I'm happy to answer any questions.
Thanks so much, Dr. Amanda Wong, for that great talk. It was really nice to see the types and then a bit of background on the stats as well as the examples that you provided. Excellent. So with that, participants, do type in your questions in the chat box. So in the meantime, I think we have questions already. So I'll just read them out for you. Okay, so a question from Faiz Ali Shah. The question is, if we have a sample of, say, 10 like measurements of BP of 10 people in different ranges, and we suppose that the mean is 120 of whole population, how can this be proved and what test is applied to it?
Could you say the 10 subjects at the BP, what that means, BP?
So I'm guessing Faiz,
yeah, if see the question.
Yeah.
Let me see, can I see the questions? Oh, well, where can I see the questions? Is it here? Sorry. Could you see? Okay, could you see it again?
Yeah, so the question is, if we have a sample of, say, 10 like measurements of BP of 10 people in different ranges, and we suppose that the mean is 120 of whole population, how can this be proved? What test is applied to it?
Oh, okay. When you do this, this is usually depends on if your 10 people only have one measurement. And then I hope I understand the question. You have the 10 subjects. You're going to compare. Your test is to compare for what? Compare to some other. So your test is you want to test your sample mean to some other values. What your underlying assumption is you think you're better, your minimum effective treatment effect. That's you need to figure out first. Then you're going to test against that. And this is 10 subject. I think usually it's I don't know. It's like if this is the phase 3 study, I don't think FDA will take this sample size in some way. 10 subjects is definitely too small. But you can do the 2 sample t-test, or depend if your end point is continuous, like I show here. But what test you're going to set up? Actually, you want to test against the minimum clinical meaningful effect there.
Thanks, Dr. Amanda Wong. Yeah. Another question. So, does adaptive design with futility stopping rules save money?
That's the one. It's common asked. I don't think so. Okay, let's put it this way. Stopping rule for futility is it's mainly for efficacy purpose. Okay, so because your drug is not working, so you don't want to treat people, with that drug. So that usually once you found the futility, as I said, actually you have already lots of people enrolled in your study already. Okay, so in that case, I don't think it's really for money saving purpose. Basically, it's for the ethical purpose. Yeah, lots of people think, oh, I stopped this earlier, I might save money. I don't think that the main purpose is the saving money.
Great, thanks, Dr. Wang. Another one here. So it is clear to find the adaptive design for phase 1 dose finding study, but for phase 2 and phase 3, what adaptive design should be chosen and under what situation?
This one is like, yeah, phase 1 is clearly because it's you're going to find MTD most of the time, but phase 2, phase 3 is it really depends on what you know about your drug. So the phase 3, let's see, like the PEMWAR I showed. So the phase 3 is if you know your drug very well, I think the Merck, they know their drug very well. So they know it's going to be successful. So they use this they use this adaptive design to get early approved and to get the drug on the market early. And if you know your drug very well, the phase 3, you can do just a simple randomization. If you don't know your drug like Merck knows their drug, so you can do a simple randomization test, go to the end to see what happened. If you phase 2, you hope you have the early accelerated approved. So the phase 2, you don't know too much, especially for the small biotech. We usually will be willing to take some complicated design because for money issue, for so for they want to know more, so that still. But I think the idea for all those clinicians and investigator is the complicated design serve multiple purpose. You think it's good, you also pay the price. You have to do lots of simulation. It's complicated. Usually the agency doesn't like it. And another is I hope, another lesson I learned, you have to go to the history data. You have to go to the based on all those, look at all the trials in the same population. Then you might want to get better information, like a better estimate of your standard deviation of your population. You can weight them together and other things. I think those are also very important, not just to go directly to find a complicated, a very fancy design. So that's, yeah, I think that's my point on how to choose those designs. You better talk to the statistician, tell them what you want, what you have, and there's lots of communication need to be done. Yeah.
Great. Thanks, Dr. Wang. We have another question from El Said Hamouda. Can you please summarize what are the benefits of adaptive design in the field of veterinary medicine?
In the field of veterinary medicine. So I'm sorry, this is, I think, in general, the design is not specified for any therapeutic area. The design is really general for but I think so I'm not the medical people. I cannot answer those questions. What the special design there? For me, I know I've done lots of oncology. I probably know oncology much better. Like an infection disease, I'm not sure. So I'm not sure what kind of the special design for that. So the design is in general depends on what you need for your study purpose. So I know CRM now is using more and more in oncology. This is a continuous dose finding method. But in other fields, probably people don't use it that much because it's not, it's not that critical. But in the oncology, you have to, the phase 1, the futility is important. We want to stop early if the drug is not working. So yeah, so I'm sorry, I cannot answer this question very well, but I hope, but in general, all the design could be used in any field.
Great, thanks, Dr. Wang. Another question, the power calculation depends on the difference between mean treatment effect and mean placebo effect. So how to choose it?
As I said, those usually choose the minimum clinical benefit effect. What do you think it's that? Or what the minimum treatment effect you want to take? The good thing is, if you calculate based on the minimum, then later your true difference actually is bigger than that. The power is always increasing. This is the monotone increasing, so you don't need to worry about that. You only calculate the smallest one, that's enough. But if you did not specify the minimum treatment effect right, if your treatment effect is smaller than that, then you could end up at the situation I gave the toy example. There are difference, but you cannot tell the difference. Okay.
Okay, thanks so much, Dr. Wang. So I'm just checking. I think we've covered all the questions now. Thank you so much to all the participants for your questions. So I guess with that, I think we're just perfect with time as well. Yeah. So perhaps with that, we can conclude the session then. Yeah, thank you so much again, Dr. Amanda Wang, and thank you so much to all the participants that have joined us. So do join us for our next webinars, and I hope everyone has a great day. Take care, and thank you very much.
Thank you for this opportunity. Bye.
Thank you.